Okay, we're live. Thank you very much for that presentation. Extremely educational, very interesting to learn about this, especially in the context of COVID-19. And certainly I've seen a lot about wildlife trade and COVID-19, and I'm sure people in our audience have as well. So we have a couple questions with regard to your review process, and then the audience, please ask any questions that you might be interested in, and I will be sure to ask those. Let me pull up a screen real quick, see if that works. So this is your actual project, or at least one of them that you referenced, and this is a public project. That means anybody in the world can go to this project. Just hide my real quick. And yeah, so the URL is sysrev.com/p/43801. When I took a look at your project, I noticed just from the sources that you uploaded 2 EndNote XML files. In your presentation, you talked a little bit about how you actually built those files. I noticed that you have, 5,000 in a first batch and then 178 in a second batch. The second one looks like an update. I'm just curious, how did you actually start this process? Was it just a couple searches on Google Scholar or PubMed or wherever, and then you learned a little bit and iterated? And also your update process. So are you planning to add more updates in the future? Yeah.
Yes, thank you very much. So yeah, as you mentioned, basically it's just a matter of the searches. So to start with it, we just did a big background literature search. So part of it was discussing with experts, just trying to identify the key literature, having chat with them and trying to figure out which are the key papers that we cannot miss if we're discussing this topic. So that gave us a list of around 100 papers that basically came out in the last 30 years or so. So just basically, yes, that was the identification of the background papers. A lot of it also came from reports that came out from WHO, OIE, FAO, WWF, WCS, etc. Like many organizations that they've been trafficked as well, which is a big one, CITES. So there are many reports and that you have the literature built into these reports. So a lot of it just was basically trying to do these reference searches within reports, within papers. And yeah, and I mentioned as well that we use Research Rabbit, which is a new platform that kind of came out at the same time that we were starting this project, and we decided to give it a go and give it a try, considering that we were just trying different things to find more literature and to build that reference base. And it was very helpful, to be honest. It really helped us to do that backwards and forward search that we needed in terms of scientific literature. But yeah, I think obviously great literature is always harder to find. You really need to do that engagement process. You really need to go into the key organizations and dig down their databases to find that. So yeah, that was the process.
Something we've done in a few of our reviews is just to look at other reviews. I don't know how many reviews have been done in the wildlife trade space, but did you use that as a way to inform, potentially missing topics, missing concepts, maybe even specific missing articles?
Yeah, so at the moment when we started the project, there were only a handful of reviews, or at least lit reviews that were on this topic. Not a lot of it was done in a single place. There were very they were mentioning different regions, different species, different aspects of trade, etc. So when we launched this, it was mostly about doing that, what you said, trying to look for those keywords, trying to look for the abstracts, trying to get a feeling about what was the field itself and what were the key aspects that were missing, for example, in terms of supply chain, in terms of species, in terms of valuation, in terms of legal, illegal, all these aspects of wildlife trade that I defined at the beginning of the presentation. So just trying to keep an eye on how that was framed in the literature.
Okay, so we have a question from the audience. If I take a look at your project, I see you have a lot of people in this project doing a lot of work here. Getting people to agree and understand inclusion criteria and data extraction can be really challenging. I'm curious how you actually educated people and trained them to do to all sort of agree on the inclusion criteria and made sure that they understood it as they move through the process.
Yeah, so this one, I would say this is one of the key aspects of this process. As I mentioned, I was very lucky to work with Kiara Keenan. She was extremely knowledgeable, and she taught me a lot to start with on how to kind of develop this process. And then once we had the main structure for the project, we connected with students from the RBC. One of the project leaders, basically, he's a professor, Richard Koch. He's a professor at the RBC school, and we invited them to participate if they were interested in the process, in that field. And we carried out a few, small workshops, you could call them, a few meetings with them, just to tell them, well, this is the system, this is the tool that we're going to use, this is how Sysrev works, and then trying to get a feeling of which were the questions that they had in terms of the themes and the field itself. So for that, we just had a Google Drive sheet where we were putting all the questions. We created Trello channel as well. We also we were using Slack to have a direct contact when questions would pop up. We'd have someone from the senior team ready to go and answer. So this was a very dynamic process, and I think really trying to incorporate all these available tools help us to have this team kind of thinking and basically just focus on the questions. Because as I mentioned from the keywords, you can find the keywords, but in many cases, the keywords they're not referring to the topic in the paper. So that was very frustrating. You would have it in the title, or you would have in the abstract, but then the paper would say something different.
So there is a tool on Sysrev called the concordance tool. To show you that real quick. I don't know if you got a chance to take advantage of it for this project. Basically, we look at your reviewers and we can see how often they agree with each other. Did you manage to leverage this and making sure everyone's on track of what labels they're answering, if they're in agreement about different things?
Yeah, so I think I didn't use it directly. But we had a lot of conversations with Kiara about the different aspects of Sysrev and the tools that were basically driving that. But I think because we had the alternative, which was the direct questions on Slack, for example, that made it more direct, because we would have a secondary or tertiary person basically just going in and discussing with the reviewer that particular article. So we kind of had a live assessment of
yeah. I mean, that sounds super ideal. Sort of like a high degree of communication between the experts and the individual reviewers. Sounds like you really squash any misunderstandings people might have. I'm curious about the map that you're building, the evidence map. First off, it's awesome that that's going to be made available. I'm curious how you're planning on making it available. Have you decided on that yet?
Yeah, so basically, because this is a UCN project, we'll put it together through the specialist group, or in a website that it will be directly linked to this particular project. So it'll be there. We're also talking with a few of the partner collaborations and organizations that they help us throughout the whole process, and they were also keen to put them in their websites. So I think obviously continuing the project, that will be ideal. But it's just one of those things that it really comes down to how much time you have to update as well and continuing doing these reviews, because the topics and the keywords that we're using with COVID-19 is just crazy, the amount of publications that you're having. So it's very hard to keep up with everything. But yeah, we're definitely trying to make it a living evidence gap map.
Nice. That was my next question. Just how will it continue to update? Sounds like that's something you guys are in the process of answering and attempting to do. So that's really cool. I wonder if you could talk about some of the specific variables that are being, identified in that map.
Yeah. So for the map, we're basically focusing on two axes. So the first one is the human disease aspect. So what are the pathogens, the microorganisms, the transmission pathway, basically all the kind of disease aspect of this. And then on the other side, we're focusing on the wildlife trade context. So what is the purpose? What is the interface? Did it happen at a very early part of the supply chain, or was it at the end? Was the contact between the trade, let's put it as the complex, and humans direct? Is it suspected? Is it confirmed? Which diagnostic tools they use to confirm, etc.? Was it epidemiological, basically knowledge about this connection, or they use PCR to find the pathogen?
Oh, cool.
So this is part of the diagnostics. Obviously, location, time, they're also part of it.
So some of them would include the experimental methods. Some of them would include disease. Some of them would include location and time.
Yeah. So these are so I would say that the main three kind of components are disease aspects, wildlife trade aspects or context, and the general information about time, location, and diagnostics.
Species too, yeah. So I wonder how we think a lot about controlled vocabularies. So like, what names are we going to use to identify a disease? People take many different approaches to that. Did you reference one of the existing ontologies for disease, or did you go ahead and invent your own disease names for this project?
So for the project, we didn't look for specific diseases, but we were just highlighting the disease that was mentioned in the project. Okay. So we didn't want to lose that type of detail. We know that, yes, we really need to make it kind of transversal and use the same vocabulary to define what we're discussing. But at the moment where we were developing the project, we thought that it would be more representative to actually capture what was being mentioned in the publications and what was being discussed, and then provide that to the researchers, to the public, etc., to make their own kind of analysis. And because we know that there are different tools where you can just kind of transform the name of either species or diseases, we thought that that would be a secondary step of the analysis.
That's a cool way to do it. Yeah. So you invent your own names, where you're not inventing them, but not using an ontology for names, but you create them quickly, and then you're able to map those to controlled vocabularies in the future. That seems like a good approach to me. Yeah. So let's see. You mentioned an iterative approach here, and not taking the time to build out robust integrations with controlled vocabularies. Seems like a good approach to me. It takes a lot of time to do that, and you want to get started. You showed a cycle in your slides where one of the last steps was the actual mapping process, and then it sort of restarted. How many times do you think you went through that cycle? I don't need an exact number, but were you sort of iterating through this over and over again and improving your labels, improving your map?
Yeah, so basically we started with a subset. With the key literature we identify, we test that out, see how it went. We use the Sysrev tool to actually see if we could capture the things that we were actually discussing and the things that we were actually asking, right? To identify the papers that would be included later in the study. So I would say between the primary search, the testing, the EPI reviewer as well, the tool itself, I think we did at least 5 or 6 rounds with different, basically different amounts of literature. But yeah, we would test it with small subset just to kind of do the extraction, do the assessment, see if the key information, the questions that we wanted to basically, the information that we wanted to extract was actually there. We were capturing that, the language was appropriate, it was clear, and then we would change it as well. So I would say, yeah, around 5-6 times.
Yeah, so quite a few. You know, reviewing articles is kind of an acquired taste. Some people can find it somewhat demotivating. Over time, you know, hopefully at least some set of people start to enjoy the process, or maybe enjoy it from the beginning. But if you're talking about iterating on reviewing articles, do you actually have reviewers sort of looking at reviewing some articles, and then, you know, you decide to change some facets of what you're extracting, and then they have to review those same articles again? Did you have any issues with keeping people motivated in that context?
Well, it wasn't about motivation at the beginning, I would say, because we were testing. So we had the thing of testing a new thing. We were trying to do something new. So we had the novelty component that is always important when we're going into these projects. And as I said, like for us, it was also a learning process. We were very happy to have students involved in the project. The students that basically help us throughout the whole project, they'll be co-authors of the papers when they come out. So you have this thing that, yes, you may get bored of the extraction or the assessment itself, but you know that then you're going to have a meeting with students. So you're going to have this kind of teaching component, and then you'll go back to it. And I think with all these, when you're very interested in the topic, you go down the rabbit hole of like, oh, so what happened next? Or you try to find a paper that is kind of related to it and do the extraction of that one. So I think there are many things that, yeah, it's just about how you take the task.
So we have a relatively small group in the audience right now, and I'm really enjoying this conversation with you, but I also want to respect your time. We have 3 minutes left, I think, for this. I'm happy to continue discussing. If you have to run, we can go ahead and end it, and I would be very thankful for everything you've done today. Would you like to keep going?
Yeah, we can keep going. I can stay for 15 more minutes.
Okay, great. That we'll time it out to 15 or so. So I am curious about that educational aspect. That is something we've seen. I think every person who's come on to speak here has talked about the educational aspect, and having students come in, learn about the process, talk about being able to collaborate and see process in these projects. That's kind of motivational for them, getting excited about AI. How many students did you actually have involved in the project?
So directly, there were 16 students.
Wow.
That participated in the primary assessment, and then in the extraction, there were 6 or 7. We wanted to keep it tight. So we basically just, we opened a call for the students that participated in the assessment, the primary assessment, and then we discussed with them if they wanted to continue to the extraction, but at the same time, we really needed people that were on top of the literature. They already had a sense of the field. They already had a sense of the difficulties when you're reading the papers. So the students that joined us to the extraction, they were amazing. They were really active. They were really engaging with the process. And as I mentioned, like just having these tools, like using Slack, for example, that gives you that kind of professional window that, yes, when you have a question, okay, I can take 5 minutes to answer this, and we move forward with the issue that we may be having at the time. So that was very helpful. And like I would say as well, having researchers, like experienced researchers doing the synthesis, and experienced researchers in terms of the expert of the field, that also helped us a lot, because it really basically gave us an extra perspective, an additional perspective on how to focus this effort.
Yeah, so that mixture of students with expert reviewers, experts in the field, seems like it has a lot of value, particularly to students. They can learn a lot. You mentioned even learning other tools, like Slack, and being able to do that in an online context. Last year, particularly, and even still now, remote work is a super relevant thing. So I'm curious about, the future of the students' research. and we have a question in the chat about the gaps from your evidence map driving future primary research. Do you see people using your evidence map to, find new places to fill it in and doing their own research and integrating that research with your work? Has that happened yet? Will you help foster that somehow?
Well, because we're collaborating, so the first part of it, like the role of the students and how we see it moving forward, some of the students that were really interested, and basically before we even finished the whole process, they were keen to adopt the tools, to use them for their master's thesis, and basically just seeing the value of synthesizing information. And I think that's a big part that we're kind of going towards now. Is just we have such a massive amount of information coming, and it's just every day, right? So I think it's very important to know how to synthesize that evidence and how to differentiate between keywords and actual relevancy of the information. So that was one part that I thought that in terms of feedback that we got from the students, that was something that for them was important. Obviously, it's a new tool. It's something new. It's something important in the field. So yeah. So I think they had that at least from their participation. That learning process that goes beyond the classroom and basically just going into a practical example of how you do the things. And then in terms of the how we see it forward. So because the project hasn't been live. It will be live in a couple of weeks. Hopefully, within the September, we'll be able to launch everything. So the idea obviously is it's open. For me, this was the first thing that we discussed. The dataset will be available. All the analysis will be there for people to go for it. We're keen to develop collaborations on this topic. This obviously, I think, COVID-19 is the second big event where we focus on wildlife trade. Obviously, we have influenza, live markets, we have SARS-1, and monkeypox, for example. And we've seen these kind of connections happening and kind of being named after different diseases. But the idea obviously is to engage more and kind of move towards the areas that we don't know and try to target those. And considering that this is an interdisciplinary field, I think that will be the value, trying to connect people from different fields, having different perspectives, having different training, and trying to connect those dots that we're missing. Because if we continue doing this kind of thing that we just do this little thing that I care about, but we lose the whole context, we lose these social dimensions, we lose the cultural background, we lose the epidemiological aspect, the virology that's behind, we're going to continue basically going down the same road.
I'm curious if you ever searched Sysrev for other wildlife trade projects. Did you do that?
Yeah, so I did it later, like after we started the project. I did it at the beginning. We couldn't find any. So we started this project last year, December. So we really launched it in January this year. So at the beginning, we searched for similar projects. I know that now there are a few other projects that are available.
When I search, I find a few being done by the user V Brooks, Wildlife and Markets, but I think those might be more of a veterinary context, just taking a quick glimpse at them. Still, wonder about opportunities to benefit from each other's projects. Yeah,
certainly. No, I think yes, for me, it's just about collaborating. Like for me, that's a part of science. Like it's not about the silos, it's about how we move ahead. So
I did have just a couple of questions about, really your topic and not so much the methods. First off, I'm kind of curious about, I guess I was aware of like wildlife trade and wet markets and that kind of thing. And I certainly, knew about zoonotic events. I don't really have a sense of how often, you're having a zoonotic event from a wet market. let's say. Did you? I mean, you sort of answered that in your presentation. Did you get sort of evidence, like a quantitative evidence of what the risk level is due to specifically wet markets and then maybe broader wildlife trade?
Yeah, so, well, in general, and I think that wasn't the focus of our research, but just from connected projects, what we're basically seeing is that this is a process issue. This is a system problem. So how are we dealing with the interface itself? So from references and from citations, it's very hard. So there are very interesting research that basically it's from it came out like a couple of weeks ago, actually. They were basically just looking at how you would have one zoonotic event or one big kind of disease event, and the literature on that topic would just skyrocket. So it's very hard to track the actual event and trying to make an assessment of the occurrence. But in general, like, we know that domestic animal trade, for example, you have disease transmission that happens very often. We know that you have the same thing from other animals. So for a matter of volumes, just for the sheer amount of animals being moved around the world, we know that domestic animal trade plays a major component. But there's a whole thing in terms of the time that we've been in contact with domestic animals, wild animals. Like in certain parts of the world, they're like the thing that you only see like on the movies, right? But for other places, it's just exactly as we see a dog or another species. So I think that's the importance. And more at the moment, I cannot say that we can quantify that exactly, like to the actual volumes. We know about volumes, we know about risk. We have an idea of the interface, that it's a high risk interface. So for example, the direct contact with fluids that I was mentioning, that's something that we know, but it's something that also happens in domestic animal trade. The thing is that we've managed to develop a system that controls for that risk at different levels, so that it puts a control point during the processing, transport, harvesting of that animal. So basically, we have this control system that is in place. And I think that's the part that we don't see when we are discussing wildlife trade in general, like the big conception of wild animals being like bats and rodents. But as I mentioned as well, we forget that wild animals are also venison, and venison trade happens in the UK. You have markets in Oxford that they have pheasants and wild boar and venison there as well. So we tend to forget that this is wild as well. Even though we are connected, we see it more often, but this is also wildlife.
A lot of the data we extract comes from clinical trials right now. So we have like a specific thing we can point at. And usually in the primary literature, that trial gets specifically referenced. You know, if you're talking about the results of a trial or analyzing the results of a trial, you'll reference that trial. So it's easy for us to sort of deduplicate the results being reported for a specific thing. I wonder if there are issues doing that in your recent research on wildlife trade. You know, if you see two primary sources, how do you know if they're discussing the same event or same data?
Yeah, so that's obviously a major issue, because what we found is that a lot of the literature just builds upon on a few papers. So you have this amplification process for certain events. So it's very hard to track down into a single event. I wouldn't say it's very hard. The thing is that it takes a lot of time, because sometimes they're referring to either the pathogen, they refer to a location, or sometimes the paper doesn't have the location, or it doesn't have the specific event, or it describes in a very general manner what it is. I would say the other problem that we kind of we found was that the disease risk assessment that we're doing now is very, I wouldn't say basic, but it really depends on the identification of pathogens in certain species. And we know that not all species or not all individuals carry all the diseases at all times. So it's very hard. That's obviously it's a very important piece of knowledge. But at the same time, we cannot just assume that that kind of species carries all the pathogens that are described for it at every single moment.
So something I was just thinking about, as a data person, is like I would really like to have a place where I could see what the geography of disease is, and where a certain disease is endemic and where it's spreading to, which is something you mentioned with climate change being a sort of a driver of emerging infectious disease. Do you know if there are sources where people are tracking disease? You also mentioned genomic surveillance, so maybe that would be something populating databases. So really two questions there. Sources of data for geography of disease, and how is genomic surveillance being populating those kinds of databases?
So in terms of spatial analysis or spatial data sets, you have the WAHIS data set from OIE. FAO also has a similar one. WHO also has a report, the SSID, I think it is. There's a big network basically that it's just reporting the outbreaks and zoonoses and emerging infectious diseases at real time, basically.
Nice.
So you have the thing, like you have it. I think the aspect that we're really looking at now is how all this comes into place, right? Like how people, how animals, domestic or wild, and how the environment come together to create this kind of interface where a disease may either spill over, or you have obviously, because it's a zoonosis, you need to have the contact between animals and humans. So that's the part that it's more straightforward. You need to have that contact component. But yeah, so also there are several papers like Jones's 2000 in the late 2000s, and you have Allen as well, Woolhouse, and so there are several papers that they've been exploring these spatial components, species richness, distribution, communities, how they relate to land use change, climate change, and other environmental factors that kind of could drive or facilitate the emergence of new diseases or the spread of existing ones.
Okay, I have so much more I'd like to ask you about all of this, but I think I risk sort of the next question I would ask would take us too far, I think. Before thanking you, any other questions from our team? It seems a few more of our developers hopped on while we have a super user here, someone who got a lot out of Sysrev. Any questions that might inform development or features or anything like that? We did ask a few already, so if there are none, that is okay. Give it just a few seconds. All right. I think we're good. So thank you so much. This was terrific. I hope we can do it again sometime. I've really learned a lot today, and it's really exciting seeing your research, all the new ways people are using these new tools and all of that. So thank you.
Thank you very much for the invitation, and yeah, definitely let's keep in touch, and I'll keep you updated when the report and the papers come out.
Okay, great. Thank you. Bye.
Great. Have a nice day.